Dr. Nolan Predigate from Dana-Farber is going to talk about characterization and proposed therapeutic exploitation of fusion RNAs in metastatic breast cancers.
All right. So on behalf of my co-authors, I appreciate the opportunity from the organizers to present this work on fusion RNAs in metastatic breast cancer. I have no financial disclosures. So firstly, this is the chromosome structure of a normal cell, and this is a spectral karyogram. You'll see that each chromosome pair is painted a different color, and it's clean and there's no colors bleeding into each other. And this is a breast cancer cell, and I think what you'll appreciate is the paint kind of has been splattered all throughout the genome, and you have many regions where chromosomes have been amplified, duplicated, and rearranged into one another. And then if you zoom in closely enough at the borders of where the colors are touching, you'll find almost a barcode that exists, a nucleotide sequence that is specific to cancer cells. And this brought us to the broad question of how can and why should we better characterize and exploit structural variant breakpoints in breast cancer. So for the first question of why should we? Well, breast cancer has a lot of them, and this was a study performed by Peacock, which showed that there was, and this is, apologies, which it's on a timer, sorry, if I can just go back. Perfect. On the left, you'll see that breast cancer has more structural variants among the most common epithelial malignancies. And importantly, on the right is a study from Nick Navin's group that showed that these structural variants occur early in a cancer's life history and can be preserved when a cancer becomes invasive. So the bottom line is that these somatic structural variants are common and are the most foundational events in breast cancer. And then they can also produce clinically meaningful fusion RNAs and nucleic acid products, and we identified an ER fusion that was present in a recurrence after therapy, and it conferred estrogen-independent activation and endocrine therapy resistance, which kind of led us to the hypothesis that fusion RNAs resulting from founder structural variant events or therapy-related selection may be under-recognized cancer-specific biomarkers and targets in advanced breast cancers. So in order to test this hypothesis, we collated data from about 466 metastatic breast cancer specimens from Dana-Farber and other publicly available data sets, and we use what we feel is a rigorous fusion finding method to both call fusions and rule out false positives. And what we ended up is with a list of high confidence cancer specific, so not found in normal fusion RNAs, and we had about a 90 to 95% validation rate in the lab. So when you look at the frequency of these high-confidence cancer-specific fusions in metastatic breast cancer, we found that basal and HER2 enriched had the highest with a median of 13 and 12 per tumor. And then we also found that, although that does not sound like a lot, when you categorize them, about 65% involve a cancer-related gene, and about 18% have in-frame kinase fusions. It's important because these are the fusions that we've generally been able to target with small molecules. Furthermore, in the top right of this plot, and I'm sorry it keeps bouncing back and forth, you'll see that we found that like the DNA level changes, breast cancer has the highest number of fusion RNAs per tumor than other common malignancies like prostate, lung, colorectal cancer, and pancreatic cancer. So looking at these fusions in more detail, they are no doubt potentially druggable. So we found 4 FGFR2 fusions that had promiscuous 3' partners. Interestingly, these were all an HR-positive disease. And then on the right, each one of the fusions had a breakpoint right at amino acid 767, which would cause the fusion to retain the entire kinase domain. We also found 2 BRAF fusions, again an HR-positive disease. And on the bottom left, we found other kinase fusions that are less immediately druggable that we're characterizing. Lastly, a fusion can produce a gain-of-function transcript, but can also cause the transcript to essentially produce an out-of-frame transcript that will be degraded. And we found recurrent loss-of-function fusions in the bottom right, like NF1, which was discussed earlier in the San Antonio Symposium, where there are point mutations that cause kind of these loss-of-function. So we argue that there may be some fusion genes that were missing or kind of knockout fusion transcripts that we need to think about the right technology to detect them, especially as we start to enroll patients into these kind of molecular trials and for these targeted therapies. So when you rank all of the fusions, all of the genes involved in fusions, this is the top of the list. We found that the most recurrent subtype enriched fusion RNA were those involving ESR1. They were significantly enriched in the LUMB classified METs, all but one. And then these ESR1 fusions have been kind of found before in case studies, but we had now one of the largest collections of ESR1 fusions, so we wanted to look at them in a little bit more detail. So at the top of this slide, you'll see kind of the estrogen receptor functional domains, and below you'll find the exons. And then that bar kind of towards the right is where all of the fusion RNAs that we found were present. And they all kind of localized right after exon 6 or right after exon 7, which is important because that causes the fusion to lose the ligand binding domain. And this is where all of our endocrine therapies bind, including tamoxifen, fulvestrant, and even the oral SERDs. So we estimate this ESR1 LBD fusion frequency to be about 5% in HR-positive treatment-refractory metastatic breast cancers. And we argue we should keep an eye on this frequency as oral SERDs kind of move more into the clinic, because those supposedly hit ESR1 mutations better, and perhaps ESR1 fusions will be a mechanism of resistance that we need to keep an eye on. We also found our collaborators at Pitt, Adrian Lee and Steffi Ostrich, have started a Hope for Others rapid autopsy program, and they also found an ESR1 fusion. Importantly, this ESR1 fusion was present in almost every metastasis from that patient, suggesting they're not subclonal events. And then we also have models that we're currently testing with this fusion. So 9 of these cases were patients treated at Dana-Farber, so we're able to look at kind of the treatment exposures and outcomes of ESR1 fusion positive disease. And on the left is a heat map of the therapies that they received. Green is endocrine therapy, blue is targeted therapy, red is chemotherapy. And importantly, you'll see that every patient before the ESR1 fusion was detected had received endocrine therapy, including all patients receiving a SERD. Importantly, 3 cases had archival primary tissue that were sequenced and no fusion was found, really suggesting that these are acquired events. And the median overall survival after the fusion was detected was 9.3 months, with only 1 patient surviving longer than a year, although this cohort is very small to make conclusive prognostic inferences. And lastly, probably one of the most important, is that 8 of 9 of these patients received targeted tumor profiling at the DNA level, and no fusion was called, so really suggesting that we need to think about the right technologies in order to detect these. And then lastly, I'll end with the kind of push for cancer genomics has been defined changes that are in multiple samples, because they likely indicate that they are a driver. But when we looked at the data, we found that over 33% of metastatic breast cancers have at least 1 fusion RNA that's expressed relatively highly, and these involve breast cancer-related genes, including TRPS1, which has been shown to be a breast cancer dependency, PIK3 pathway members, ERBB2 fusions, etc., which kind of raises the question of are these N of 1 drivers, these patient-specific, or are they passengers, and where do they arise? Regarding the question of when they arise in therapy, we were able to analyze the Aurora cohort, which has a couple rapid autopsy cases, and we importantly found that these highly expressed fusion RNAs can be expressed in every single cancer tissue from an individual patient, suggesting that they may be kind of more homogeneous targets than what we are currently targeting. So this leads to a question that we're excited about. How can we really leverage advances in gene therapy to build medicines that can exploit these nucleic acid targets directly? And can we move past kind of this protein-centric cancer medicine and move towards targeting nucleic acids? And the FDA is debating on whether or not to approve the first CRISPR therapeutic, I think, today in sickle cell disease. So we think now's the time to think about how we can apply some of this technology to cancer. So in summary, patient-specific, highly expressed fusion RNAs are present in over 1/3 of metastatic breast cancers. We have low-frequency recurrent gain and loss-of-function fusion RNAs that likely drive therapy resistance in metastatic breast cancer. Some are potentially druggable, such as FGFR family and BRAF fusions, and likely missed with current testing standards. 5' ESR1 fusions that cut off the ligand binding domain are some of the most recurrent fusion RNAs in metastatic breast cancer, with a frequency of 5%. These are acquired in the treatment refractory setting. They can be widely expressed in multiple metastatic sites of an individual patient, and we feel that, with the breakthrough advances in gene therapy and nucleic acid targeting, we could potentially think about outside-the-box therapies that target these cancer-specific breakpoints. And I apologize for the slides. They were running faster than I was, but I thank everyone on this slide, especially my mentor, Dr. Todd Gullivan.